the last talk in the last session. I hope that I will keep it on time. I'm cardiac electrophysiologist and physiologist, so I don't know whether this topic, how this topic will help to the personalized medicine, but this is our preliminary work, so it's open for the comments and discussion. So we know that the heart is a pump, so it doesn't matter what the status is, as long as it pumped the blood out, then it done its job. And the function of the heart is mediated through the function of the single cardiomyocyte. So we know that the heart disease or cardiovascular diseases remain to be the leading causes of human mortality, suggesting that there are some aspect of the heart functions that we don't know. Mechanistically, there are many questions that has been unsolved. Currently, in vitro experimental studies, we use two techniques to identify or examine the function of the heart. One is to isolate the heart, and then using the Langendorf system to look at the pressure changes. On the other hand, in single isolated myocytes, we can actually directly measure contractility of the myocyte. And the topic I'm going to, or the results I'm going to present today is mainly through this method. In cardiac physiology field, it is all known that the fundamental mechanisms that underlying the myocardial function is cardiac excitation contraction coupling. So I separated this into two parts. One is the trigger part of the excitation contraction coupling, and then the execution part of the excitation contraction coupling. So we all know that when the action potentials propagate to the myocyte, it can depolarize the membrane potential and then triggers the L-type calcium channels to open, so that there are small amount of calcium that get into the cells and trigger a lot of calcium to release from the calcium store, which is a sarcoplasmic reticulum. And this increase in intracellular calcium can stimulate or bind to the calcium binding proteins in thin filaments, so that it can initiate some conformational changes, and also the association between the thin and thick filaments, so that it can contract. And conversely, when the calcium is dissociated from these calcium binding proteins and the calcium reuptaken back into the calcium store, that the intracellular calcium is reduced, then it will trigger relaxation. So for the last 2 or 3 decades, there has been suggested that this calcium handling mechanism is the predominant mechanism to understand the cardiac contractility. However, because the myofilament accounts for the majority of the volume in the myocardium, therefore this is actually playing quite an active role in the contraction and the relaxation. And there are already a lot of work actually going on to understand these mechanisms. And recent concept is that there are also crosstalks between the myofilament and the intracellular calcium handlings, and we get more insight into the cardiac contractility. And there, apart from that, there is also an active contribution of mitochondrial metabolism that is contributing to myocardial function, which I think is not, it's the field that hasn't been understood correctly or clearly. So if we go back to have a look at the cardiac metabolism, as we all know that this glucose as the one of the main metabolic substrate that it can produces cellular ATP, And this is what we all understood. However, the fact that hasn't been understood by many of the cardiologists or cardiophysiologists is that the fatty acid is actually the main predominant metabolic substrate for ATP production in the heart. And in fact, there are more than 70, in average of over 70% of ATP are produced through fatty acid metabolism comparing to glucose metabolism. On the other hand, in diseased heart, there are dysfunction of this fatty acid dependent metabolism in cardiomyocyte, and reversely, there are heavily glucose dependent metabolism that are contributing to the maintenance of ATP and therefore contraction. However, over the almost half of century that for the cardiac physiology experiments, most of the experiments has been done under the conditions where the glucose as the main or only metabolic substrate that has been used, and also the concentration of glucose has been very high. It's up to 10 to 15 millimolar, so therefore this is highly not physiological. And we know that in the blood supplies that contains the metabolic substrate that feeds the myocardium or the cardiac cells, there are glucose as well as a number of fatty acids that actually can be uptaken by the myocyte. Therefore, there must be some differences that we haven't been understood about the cardiac metabolism and its contribution to cardiac function just because of these different conditions that we have been used. So this seems a quite simple question, but I think this is quite important because this is the area of a field that we haven't been explored in the past decades. And on the other hand, there are some remodelings of the plasma or serum fatty acids that under disease or stress conditions, for example, these are the group of patients that has been examined in the serum level of fatty acids after acute myocardial infarction just under 6 hours. And they have been found that the serum level of fatty acids are immediately increased with stress. And in animal models, it has also been shown that by rapid pacing of the left ventricular chamber and induce this cardiac dysfunction, you can already see that there is a very quick increase of a serum level of fatty acid that and concomitantly there is no uptaken or less of the usage of these fatty acids in the myocardium, suggesting that there are lots of remodelings going on, therefore the metabolism can affect the cardiac physiology and the pathology. So, so to summarize of this phenomenon is that at the moment, under physiological conditions when the myocardium should use fatty acid to produce ATP and accomplish the contractility. However, we are not giving any fatty acids, therefore, in a way that we are disabling the beta oxidation of the cardiomyocytes. On the other hand, when there are some remodelings already happened in the serum level of these patients compared to the healthy subjects, that we are still using the same type of solutions that analyze this function. Therefore, we just aim to understand these differences and to see whether if we supplement fatty acids to the normal thyroid solution that we have been using for quite a long time, and what happens to the cardiac function, and what will happen if the myocardium are under stress conditions. So just the screen for the effects of this fatty acid, we treated with one single dose of, one single type of unsaturated long-chain fatty acid, palmitic acid, because this has been mostly documented because of the unhealthy food and all of the nutrient issues that has been associated with this type of saturated long-chain fatty acid. And we found that when we treat the palmitic acid at different doses, the myocyte contractility progressively increases without affecting the relaxation parameters, suggesting that this fatty acid actually play a role in myocyte contractility. And on the other hand, if we give stress to the myocyte, we infuse this angiotensin II to produce the hypertension model in the rats, we found that the effect of fatty acid actually abolished. So suggesting that there are changes in fatty acid and metabolic substrate utilization in these two different models. And we know that there are, under stress conditions, this beta oxidation are the targets of angiotensin II. Therefore, it's possible that these type of treatments are necessary to differentiate between the normal cardiac physiology and the physiology. Because if we don't supply this fatty acid, you will see that there will be no change in the contractility. It will come out with the conclusion that the myocyte contractility is not changed. However, if we stress, if we give the fatty acid, treat it with a fatty acid, then you will immediately see the differences between the two groups. And we can actually carry on with a mechanistic study. And palmitic acid obviously is not the only fatty acid that can function as a metabolic substrate. There are many different types. We did some literature search and found that in the rat plasma and in the serum, there are at least 3 types of fatty acid, which are supposed to be the predominant components, and also there are some other metabolic substrates like pyruvate, lactate, and carnitine. That so we, according to that literature that we gave, we supplied these metabolic substrates to the normal thyroid solution, and we reduced glucose to the physiological level, and conducted this study. And as you can see here, that with normal thyroid solution, if we give, this is the contractility that we can see under normal thyroid solution conditions. However, if we apply these metabolic substrates, the contractility significantly increase. And if we give some stimulation with a beta-adrenergic stimulation, then contractility further increases. And as you can see here, both the contraction and the relaxation are significantly increased by this nutrient supplementation. What is striking is that we found that with the metabolic substrate, the myocytes start to have abnormal contractions. As you can see, that they start to have spontaneous contractions at the diastolic level, and the incidence of this arrhythmia, like myocyte abnormal contraction, increases with beta-adrenergic stimulation. And these abnormal contraction seem to be related to the mitochondrial activity, especially the fatty acid transferring, one of the key enzymes that contributing to fatty acid or beta oxidation metabolism. If we use this carnitine palmitoyl transferase 1 inhibitor, renolazine, which has been designed to treat the myocardial dysfunction, especially in the ischemia conditions. If we treat this renolazine, we found that the contractility is not affected. However, it significantly reduces the arrhythmia genesis in these myocytes. However, this is not due to the mitochondrial overactivity, therefore induced oxidative stress, because if we use the antioxidant NAC, it didn't reduce the arrhythmiogenesis. However, it actually reversed it. So this data overall is suggesting that mitochondria is important in arrhythmiogenesis as well. So what is the basis of increasing contractility? And as I said, that because EC coupling is the basic scheme for the contraction, to understand the mechanism underlying the contraction, therefore we looked at action potentials. We found that action potential duration are significantly prolonged, maybe can underlie some EAD or DAD phenotype. So we went on to look at the ion channels. Those are responsible for this action potential duration prolongation. And as you can see that, to our surprise, the L-type calcium channels activity are not affected by metabolic substrate supplementation, and also the sodium calcium exchanger. Both of the L-type calcium channel and the sodium calcium exchanger are responsible for the plateau phase of the action potential duration. However, these two parameters are not affected by fatty acid or metabolic substrate. On the other hand, intracellular calcium handlings are significantly affected by this, and as you can see that with metabolic substrate supplementation, the calcium change and amplitude are significantly increased and increased further with the beta-adrenal stimulation. And similar to what we see in the contraction, there are some spontaneous calcium release, diastolic calcium release under these conditions. So if we look at this, these phenomena more carefully, we found that the peak calcium amplitude, so the total amount of calcium, is significantly increased, and this is due to the calcium release through the sarcoplasmic reticulum, and this circa is responsible for the reuptake of calcium. So we would expect that if the calcium is increased, there are some slower reuptake of calcium through circa. However, you can see that the circa activity is significantly increased, suggesting that there are some increasing in the circa activity, and therefore increase in the calcium release, because the peak time of calcium release. So if we go back and see, there is the dome shape. The peak time of calcium release is significantly increased with the NF, and it's increased further with isoprenaline treatment. So these data suggest that the circa function, or sarcoplasmic reticulum function is normal. We have just shown that the L-type calcium channel or the sodium calcium exchanger, which are responsible for intracellular calcium increase, are not affected. We have just published recently suggesting that this myofilament can be another important source of intracellular calcium, and if there are some changes in myofilament calcium sensitivity, then it will feed to the intracellular calcium and can contribute to the myocyte contractility and also the arrhythmogenesis. Therefore, we went on study the myofilament calcium sensitivity under these conditions, and we found that, as you can see at the lower panel, that myofilament calcium sensitivity are significantly reduced, and maybe suggesting that this is the primary mechanism for increase in intracellular calcium and therefore arrhythmogenesis. So we don't know what is the mechanism actually leading to this, but we have some preliminary results showing that under these conditions, with fatty acid supplementation already, that there are some changes on the insulin receptor signaling. So the tyrosine phosphorylation of insulin receptors are significantly reduced with the fatty acid treatment only, and together with insulin receptor substrates, suggesting that there are some changes in the intracellular signaling that leading to the metabolism changes. And downstream of this insulin receptor signaling, the enos phosphorylation, which is an active site of enos phosphorylation, or also significantly reduced with the fatty acid supplementation, suggesting there is a nitric oxide deficiency that may affect to the cardiac physiology. And we have results showing that under normal conditions, insulin signaling can have anti-adrenergic effect under these conditions. However, if we abolish enos with the L name, the effect of insulin are significantly impaired. So therefore, there is still positive inotropic effect with the isoprenaline. However, in NF conditions, it doesn't matter whether we infuse insulin or with or without enos inhibition, the effect of insulin has abolished, suggesting that there are the insulin signaling has been disabled, and also with the nitric oxide inhibition can significantly increase arrhythmogenesis. So therefore, this insulin receptor and the enos-dependent signaling may contribute to fatty acid-dependent arrhythmogenesis under these conditions. So based on these studies, although I told you that this is quite preliminary studies, that metabolic substrates, especially with the fatty acids, can increase myocardial contractility. And this effect is exerted by affecting myofilament calcium sensitivity and also intracellular calcium handling. And to our surprise that we found the metabolic substrate, we thought this can be the physiological conditions, but it can induce arrhythmia under these in vitro conditions, so especially under beta-adrenergic stress conditions. So this is, in a way, that in vitro model of a metabolic substrate, mimicking metabolic substrate. Therefore, we can use this model to study that mechanism further. And mechanistically, although we haven't fully explored this mechanism, that insulin and nitric oxide-dependent signaling pathway and the dysfunction of this may contribute to this phenomenon. So I would like to conclude with thanking the contributors and the collaborators for this project. Thank you very much.
Thank you, Dr. Zhang, for a very nice presentation. This paper is open for discussion. Any questions? I think you satisfied all their curiosity. Thank you.
Thank you.
Thank you very much.